You're taught from day one of biology class that, for an infectious disease to spread, it needs a blueprint.
Right, like some kind of genetic material.
Exactly. Needs genetic material. I mean, bacteria have DNA. Viruses have RNA or DNA. Even parasites, fungi, they all carry a genetic code.
Yeah, a code that tells them exactly how to replicate, how to invade your body, and, well, how to survive.
Right. It's the engine of replication. So if I told you there was a deadly infectious agent that had zero DNA, zero RNA-like, no genetic blueprint whatsoever, and yet it was 100% fatal and essentially indestructible, you'd probably tell me I was pitching a science fiction movie.
Oh, totally. I mean, it sounds impossible. It basically breaks the fundamental rule of infectious disease.
But it's very real. And today we're doing a custom deep dive into a stack of incredibly detailed sources. We've got clinical overviews from the CDC, a comprehensive medical review from Stat Pearls, some patient guidance from Johns Hopkins Medicine, and a really fascinating veterinary science review from the Open Veterinary Journal.
It's a heavy stack of reading, for sure.
It really is. And our mission today is to look at exactly that. The fascinating and frankly terrifying world of prion diseases, specifically Creutzfeldt-Jakob disease and bovine spongiform encephalopathy. Okay, let's unpack this. How on earth can an infectious agent possibly exist without any genetic material at all?
It's a brilliant question. And, answering it requires us to completely rethink what an infection actually is.
Okay.
The discovery of this agent completely upended the biological sciences. It's called a prion, which stands for proteinaceous infectious particle.
Proteinaceous infectious particle. Got it.
And that is all it is. It's just a single protein. To understand why this is such a paradigm-shifting concept, we really have to look at how normal proteins work in your body.
Like the ones we already have.
Exactly. Proteins are essentially long chains of amino acids, right? And they fold into very specific three-dimensional shapes. The shape of a protein entirely dictates its job. So what we are dealing with here is a perfectly normal protein one sitting in your brain right now that simply folds the wrong way.
Wait, so if this is just a normal protein already in my brain, what is it actually supposed to be doing? And what triggers it to become a threat?
Well, in the medical literature, this normal cellular prion protein is called PRPC.
PRPC.
Right. And it's heavily concentrated on the surface of cells in your central nervous system. In its healthy state, its structure relies on things called alpha helices.
Alpha helices. I'm trying to picture that. Is it like a loose, flexible, slinky kind of shape?
That is a perfect analogy, actually. Yeah, the normal protein is like a flexible slinky. It's soluble, it does its job, which researchers think is related to protecting nerves or helping with cell signaling. And when it wears out, your body's enzymes just easily break that slinky down and recycle the parts.
Makes sense. Normal biology at work.
But the disease process kicks in when that slinky undergoes a radical structural change. It misfolds into the pathogenic version, which is known as PRPSC.
And what's the CES stand for?
It stands for scrapie. That was the very first prion disease ever identified, and it affected sheep.
Okay, so what does the misfolded version look like? If it's not a bouncy slinky anymore, what is it?
Well, those flexible alpha helices convert into something called beta-pleated sheets. So if the normal protein is a flexible slinky, the misfolded beta sheet is like, think of a stack of tightly interlocking Lego bricks.
Oh, wow.
Yeah, flat, rigid, and completely inflexible.
And I imagine the body's natural recycling enzymes, like those chemical hammers meant to break down the slinkies, they just bounce right off those interlocking Legos.
They absolutely do. The body just cannot digest them. But what makes this truly an infectious agent is the domino effect.
The domino effect.
Right. When this rigid, misfolded Lego brick comes into physical contact with a normal slinky protein, it acts as a corrupted template. It literally physically forces the normal protein to refold into the rigid beta sheet shape.
Are you serious? Just by touching it?
Just by touching it. So now you have 2 misfolded proteins. They bump into 2 more, creating 4, then 8. It's self-propagation without reproduction. There's no DNA dividing here. It is purely a structural chain reaction spreading through the tissue.
But wait, I have to push back here. If it has no DNA to replicate and no real biological drive, how does it physically move from cell to cell? I mean, a chain reaction in one cell is bad, but how does it infect its neighbors?
It's wild. But the sources outline 2 primary delivery methods, and they show how prions basically hijack the body's normal communication networks.
Okay, what's the first one?
The first involves exosomes. Cells naturally pinch off little bubbles of their own membrane to send chemical messages to other cells.
Like a microscopic care package?
Exactly. But a cell that's overwhelmed by prions will accidentally bundle some of these misfolded proteins into an exosome and just float it over to a neighboring cell.
Oh, man. A Trojan horse.
Pretty much. And the second method uses tunneling nanotubes.
Nanotubes. Now we really are back in sci-fi territory.
I know, right? They are incredibly thin, temporary membrane bridges that naturally form between two cells. The prions literally move across these physical bridges to invade the neighbor. It's just a stealth invasion.
Which is terrifying. And the sources say these things persist in soil or, on stainless steel surgical tools. How does a dead protein survive outside the body if it's not a living organism trying to survive?
What's fascinating here is that its survival isn't an active biological process. It is purely chemical resilience.
Because it's just a shape.
Exactly. Because it has no genetic material to destroy and no cell wall to burst, standard medical sterilization does absolutely nothing. I mean, you can soak a prion in 70% alcohol.
Wait, rubbing alcohol doesn't kill it?
Not at all. You can bathe it in UV radiation or even put it in a standard medical autoclave at 121 degrees Celsius, and it will remain perfectly infectious.
That is insane.
The tight stacking of those beta-pleated sheets makes them chemically impenetrable. To actually inactivate a prion, you need incredibly harsh conditions. We're talking heating it to 134 degrees Celsius for up to 18 minutes while simultaneously dousing it in concentrated sodium hydroxide or bleach.
So you're not trying to kill a bug. You're basically trying to chemically melt a microscopic piece of armor.
Precisely. You have to melt the armor.
So if these indestructible Lego bricks are just spreading through cell tunnels and surviving literally everything, what actually happens when they pile up inside a human skull?
This kind of brings us to the human cost, right? Classic Creutzfeldt-Jakob Disease, or CJD.
Yes. Because the body's normal recycling enzymes can't digest these beta-pleated sheets, the prions just clump together inside the neurons. As they accumulate in these massive plaques, they disrupt the cell's internal machinery, and eventually the neuron just swells up and dies.
And when you look at this under a microscope, the pathology is pretty striking. The sources note that the dying neurons leave behind these empty vacuoles, basically holes that are like 20 to 40 micrometers wide. Right. They are literally turning the brain's gray matter into a sponge. That is where the medical term spongiform encephalopathy actually comes from.
And while the neurons are dying and leaving these microscopic holes, the brain's support cells, the astrocytes, realize something is terribly wrong, right?
Yeah, they go into absolute overdrive trying to clean up the damage. And this leads to astrocytic gliosis, which is essentially massive inflammatory scarring throughout the brain tissue.
And that physical destruction translates to some really horrifying symptoms. The sources describe a rapidly progressive dementia. We're not talking about the slow cognitive decline you might see in Alzheimer's over years. This is incredibly fast.
It's devastatingly fast.
Yeah.
Patients develop memory problems, confusion, and behavioral changes. And because the physical structure of the brain is disintegrating like a sponge, the actual hardware required to form a word, or hold a memory, or even move a leg is simply melting away.
The speed of the deterioration is a hallmark of classic CJD, isn't it?
It is. Patients often experience myoclonus, which are these sudden, involuntary jerking movements of the muscles. And tragically, these severe jerks are often triggered by something as mundane as a sudden loud noise or a bright light in the room.
Wow. Just a loud noise.
Yeah. As the brain loses its fundamental architecture, the patient loses the ability to walk, to speak, and eventually they slip into a coma. The prognosis for classic CJD is universally 100% fatal.
100%.
Yes. And the median duration of the illness, from the onset of symptoms to death, is just 4 to 5 months.
Just 4 to 5 months. That is brutal. So to understand who gets this, the literature breaks classic CJD down into 3 origins. What are we looking at there?
Well, the first is the familial or genetic type, which makes up about 10 to 15% of cases. That's where a person inherits a mutation in the PRNP gene, which simply makes their normal slinky proteins naturally more prone to misfolding into Lego bricks over time.
Okay, so it's inherited. What's the second?
The second is the iatrogenic type. This accounts for less than 1% of cases. Iatrogenic just means it was acquired through a medical procedure.
And reading about those iatrogenic cases is deeply unsettling. Because prions are so resilient to normal autoclaves, patients in the past actually contracted CJD from contaminated neurosurgical equipment.
Yes, unfortunately.
The sources also mention transmission through duramater grafts, which is the membrane surrounding the brain, and from human growth hormone that, before 1978 was harvested directly from the pituitary glands of human cadavers.
It is a really tragic chapter in medical history. Doctors had no idea that their standard rigorous sterilization protocols were leaving behind this indestructible protein. But once the medical community realized what was happening, protocols for handling nervous system tissue were drastically changed, and artificial growth hormones were developed. So those iatrogenic cases are now exceedingly rare.
Which leaves the third type, sporadic CJD. This accounts for the vast majority, right?
Yeah.
About 85% of all cases. And it mostly strikes people in their late 60s. The sources say this happens randomly for absolutely no known reason. The protein just spontaneously misfolds. Here's where it gets really interesting. If that's true, how can doctors possibly predict or stop it?
The hard truth is, right now we can't.
We can't do anything.
No, because sporadic CJD is a spontaneous structural error at the molecular level. There's no virus to vaccinate against, no bacteria to target with antibiotics. We cannot predict whose proteins will suddenly collapse into a beta sheet.
That's terrifying.
It is, but it is vital to keep the statistics in perspective for anyone listening. While the mechanism sounds terrifying, sporadic CJD is incredibly rare. We're looking at an estimated rate of only 1 to 2 cases per million people per year.
Oh, okay. Yeah.
In the entire United States, that translates to about 350 to 600 cases annually. It's a catastrophic biological lightning strike, but a very rare one.
Okay, so if 85% of these cases are just sporadic lightning strikes, what the heck happened in the United Kingdom in the 1980s? Because that wasn't a lightning strike, that was an absolute explosion. We need to talk about mad cow disease.
That is a crucial pivot. Classic CJD, as we just discussed, arises spontaneously or genetically. But the outbreak in the 1980s was an acquired infectious disease directly tied to bovine spongiform encephalopathy, or BSE.
And according to the Open Veterinary Journal source, this was essentially a man-made disaster driven by industrial agriculture.
Yes, unfortunately.
To boost protein in cattle feed, the industry was using something called meat and bone meal. They were taking the leftover offal from slaughtered cattle and sheep, including brains and spinal cords, rendering it down and basically feeding it back to cattle.
It was an unintended recycling program for prions. If a single cow or sheep had a spontaneous prion disease, its tissues went straight into the rendering plant.
And since we know prions survive extreme heat, the rendering process didn't kill it.
Exactly. The infectious agent wasn't destroyed. It was concentrated, packaged, and fed directly to hundreds of thousands of other cattle. This caused a massive epidemic. Over 200,000 cattle were infected.
200,000.
Yeah, the economic impact was staggering, with billions of dollars lost and export markets completely collapsing.
But the real tragedy hit when it crossed the species barrier to humans, right? People were eating contaminated beef products that contain nervous system tissue. And this led to the emergence of variant CJD in humans. The sources point out that variant CJD behaves very differently from the classic sporadic version.
It really is a distinct clinical picture. While classic CJD typically affects older adults, variant CJD hits much younger people. The median age of death for VCJD is just 28 years old.
That's so young.
It is. And the disease course is also agonizingly longer. It lasts about 13 to 14 months rather than 4 to 5, and it often presents completely differently at the start. Well, instead of immediate memory loss, VCJD frequently begins with severe psychiatric symptoms. So deep depression, anxiety, extreme withdrawal, and painful sensory sensations. And this is long before the severe neurological decline and dementia actually set in.
It's just wild to me that eating a completely different species can trigger this. But it actually wasn't the first time an orally transmitted prion disease was documented, was it? The sources dedicate quite a bit of time to Kuru, which affected the 4 people of Papua New Guinea.
Right, Kuru is a foundational piece of prion research. The disease was spread through ritualistic cannibalism. When a tribe member died, it was a cultural practice to consume the body as a sign of respect and mourning.
And the prions were concentrated in the brain.
Yes, the women and children were typically given the brains and internal organs to eat, and those tissues were just teeming with prions if the deceased had died from the disease.
So it hit them the hardest.
Exactly. The epidemic heavily decimated the women and children of those villages. It directly proved how incredibly potent the oral transmission route is for nervous system tissue.
Which naturally makes me wonder about vulnerability across other species. If cows can eat tainted feed and humans can eat tainted beef, can any animal get this? Like, what about our pets?
Species barriers in prion diseases are highly complex. Some animals seem quite resistant. For example, there's no evidence that pigs or chickens can naturally contract the disease from oral transmission, likely due to structural differences in their own cellular proteins.
Okay, good for the pigs and chickens.
But our pets are not immune. During the BSE outbreak, Domestic cats actually contracted feline spongiform encephalopathy, or FSE, from eating commercial cat food that contained the same contaminated bovine brain and spinal cord tissues.
Wow. So the contaminated feed literally poisoned the whole food web.
It did. But if we connect this to the bigger picture, here's the silver lining. The science directly solved the crisis.
How do they do that?
Well, once researchers identified the transmission route, governments globally implemented strict feed bans. They completely stopped the recycling of ruminant proteins back into animal
They
feed.
cut off the source.
Exactly. That single, science-based public policy successfully halted the BSE epidemic. It proves that understanding the underlying biology can save countless lives and rescue entire agricultural economies.
Which is a massive relief. But it brings up a very practical and kind of terrifying problem for neurologists today. With an infectious agent that hides this well one that triggers no antibody response, no white blood cell spikes, and initially just mimics other forms of dementia, how on earth do doctors find it without drilling into a patient's skull for a brain biopsy? Because doing a biopsy on a CJD patient sounds like a great way to contaminate an entire surgical suite.
Brain biopsies are indeed the absolute gold standard for confirmation, but you're right, they are highly invasive, very dangerous, and heavily avoided unless doctors are desperately trying to rule out a treatable condition.
So what do they do instead?
Neurologists rely on a combination of advanced surrogate markers to essentially hunt the invisible agent. When a patient presents with rapidly progressive dementia, the first step is ruling out other rapid onset causes, like autoimmune encephalitis, multiple strokes, or central nervous system infections.
And to look specifically for CJD, they use MRI scans and EEGs. The literature mentions radiologists looking for cortical ribboning or the hockey stick sign. What are they actually seeing there?
They are basically seeing the battlefield left behind by the prions. An MRI measures water and tissue density. In sporadic CJD, cortical ribboning shows up as a distinct brightness along the folds of the brain's gray matter.
And what about the hockey stick?
In variant CJD, the hockey stick, or pulvinar sign, appears as bright spots in the deep structures of the brain called the thalamus. That brightness on the MRI isn't the prion itself. It's the intense inflammation, the spongiform vacuole formation, and the massive astrocytic scarring we discussed earlier. It literally shows where the brain is actively degenerating.
They also run an EEG to measure electrical activity, right?
Yes. And in the later stages of CJD, the EEG will often show periodic sharp wave complexes. Because the structural integrity of the brain is collapsing, the electrical signals begin to physically misfire in a very specific repeating pattern that is highly supportive of the diagnosis.
The doctors can also test the cerebrospinal fluid, right? The fluid surrounding the brain and spinal cord. The sources mention testing for 1433 or tau proteins, but the text is very careful to point out that these aren't actually prions.
Correct. The 1433 and tau proteins are simply markers of rapid neuronal death. Think of them as the smoke from a fire.
Ah.
When brain cells are destroyed quickly, they rupture and spill these structural proteins into the spinal fluid. So an elevated level tells you that the brain is degenerating at an alarming rate, which is a major red flag for CJD, but it isn't definitive proof of the prion itself.
But the medical field has a new weapon. The Stat Pearls review details a cutting-edge test called RTQIC, real-time quaking-induced conversion.
Yes.
This actually detects the prion itself, but how do they do that if the amounts in the fluid are so microscopically small?
RTQIC is a brilliant piece of diagnostic engineering because you cannot easily detect the incredibly sparse, misfolded prions in spinal fluid. The test purposefully amplifies them.
Oh.
They take a sample of the patient's fluid and mix it in a test tube with a massive amount of normal, lab-grown slinky proteins. Then they literally shake the test tube. That's the quaking part.
Oh, so they are intentionally speeding up the Domino effect.
Exactly that. If there is even a single misfolded Lego brick in the patient's sample, the violent shaking causes it to crash into the normal proteins, forcing them to misfold.
Wow.
They just let the chain reaction run wild.
Wow.
Yeah, within hours, the test tube rapidly fills with misfolded beta sheets, which are designed to bind to a specific fluorescent dye. If the machine detects the glow, you have a definitive positive diagnosis. And what is truly revolutionary is that scientists have found you can perform this test not just on spinal fluid, but using olfactory epithelium brushings.
Meaning they can just take a swab high up inside your nose near the olfactory nerve to get the sample.
Yes. The olfactory nerve provides a direct pathway to the brain. It takes a diagnosis that used to require a dangerous surgical brain biopsy and makes it as minimally invasive as a deep nasal swab. It is a staggering leap forward in diagnostic accuracy and patient safety.
So what does this all mean for you listening right now? Are you taking a gamble every time you eat a burger or go hunting? Because we've talked about mad cow, we've talked about indestructible proteins in the soil. It can sound like a total minefield.
It is entirely understandable to feel that way. However, I can confidently reassure listeners that commercial beef is incredibly safe today. The global feed bans, combined with strict surveillance and the mandatory removal of specified risk materials, meaning slaughterhouses, completely remove the brains, spinal cords, and certain tissues from the human food chain, have effectively neutralized the risk of BSE.
What about blood donations? Because the sources mention there are still restrictions for people who lived in the United Kingdom during the 80s and 90s.
Right. Because variant CJD can be transmitted through blood transfusions, many countries still maintain blood donation bans for individuals who resided in the UK during the height of the BSE epidemic. The incubation period for prions can span decades, and we still do not have a routine, high-throughput blood screening test for asymptomatic carriers. But this raises an important question regarding another ongoing risk mentioned in the sources: chronic wasting disease, or CWD.
Right, CWD. This is a prion disease that affects deer, elk, and moose. And unlike BSE, which only spread through contaminated industrial feed, CWD spreads naturally out in the wild.
Exactly. CWD prions are highly concentrated in the saliva, urine, and feces of infected animals. Because prions are so environmentally resilient, they bind to soil and persist in the forest for years, infecting other herds as they graze.
So it's literally in the environment.
It is. Now, while there are currently no confirmed cases of CWD jumping the species barrier to humans, the risk is not zero. If you hunt, it is deeply important to have your deer or elk meat properly tested by state wildlife agencies before you consume it. Never handle or eat meat from an animal that looks sick or acted strangely in the wild.
It's all about being informed and taking the right precautions. So let's just recap this incredible journey. We started with the startling realization that a simple microscopic protein with no genetic blueprint, no DNA, no biological drive to survive can create a massive domino effect of destruction. We saw how this indestructible Lego brick turns gray matter into a sponge, causing diseases like sporadic CJD that strike randomly and with devastating speed. We traced the agricultural missteps that led to the mad cow epidemic and how global public policy successfully stopped variant CJD. And finally, we explored how brilliant diagnostics like the RT-QIIC test are giving doctors a window into the brain using just a nasal swab.
It really is a remarkable story of medical detective work. And as we conclude, I want to leave you with one final mind-bending thought from the research.
Lay it on us.
We know that prion diseases require the normal, healthy cellular protein to exist, so the corrupted prion has something to misfold. The domino effect requires dominoes. Well, scientists engineered mice that were completely stripped of their normal cellular prion proteins. They knocked out the gene entirely.
So they had no normal slinky proteins at all. What happened to them?
They lived relatively normal lives. It turns out the body might not strictly need these normal prion proteins to survive. Furthermore, when scientists depleted these normal proteins in mice that were already infected with the prion disease, it actually reversed the early spongiform damage in their brains.
Wait, if you just remove the normal proteins, the corrupted prions have no dominoes left to knock over. The chain reaction simply starves.
Precisely. Which leads to a fascinating theoretical question. Could the ultimate future cure for prion diseases be a genetic switch that simply turns off our body's natural prion production entirely, effectively removing the fuel for the fire before the spark even hits?
Imagine that. Genetic deletion as the ultimate cure for an unstoppable disease. That is definitely something for you to ponder the next time you think about the delicate microscopic folding happening inside your own cells. Thank you for taking the plunge into the sources with us. Keep questioning the rulebook, and we'll catch you on the next deep dive.